that will be the material for the third exam. So after that, we're going to go to evolution, okay? And that'll be on the fourth exam. So let's review a little. In terms of, remember, a chromosome, a bunch of DNA wrapped around some proteins, right? So you got hundreds of thousands of genes. So things can happen to this spontaneously. It's not inherited, right? So what's one disorder we talked about? Everybody remember?
Deletion.
Okay, great, awesome. Deletion. So deletion is where a piece of it just breaks off and is lost forever. Okay, and when any of these happen, you get a disease usually. What are the deletion diseases we talked about? Do I remember? So like you have a deletion of chromosome 5.
Cri-du-chat.
Deletion of 5, right? So you have cognitive delay. The voice box sounds like the mewing of a cat, right? Your developmental problems. The other one is Williams syndrome, which is deletion of chromosome 7. Very good. Okay, so another one I'm going to talk about is this one here, D, called translocation. Okay, so these are two non-sister chromosomes. They have different numbers. So let's say this is 9, this is 22. What's happening between the two? Anybody tell me what's happening? So let's say these are two different number of chromosomes. What's happening in a translocation event? What do you see happening? Okay.
It's a translocation, I guess.
So like what's happening in your words? Chromosome 9 or 22 here?
They got like mixed up.
Yeah, so pieces are mixed up, right? They exchange pieces. Okay, so this is an event that can happen in real time too. Okay, so as an example, say this is chromosome 9, and this is chromosome 22. Could be other numbers too. Let's say there's a gene on 9 called ABL, and there's a gene on 22 called VCR. Okay, both these genes are involved in mitosis, cell division. So what happens in a translocation event between these two guys is you get an exchange of pieces, and 22 is the important one to watch here. You get a new 22 where the ABL and the VCR are brought closely together. So they're brought together, they're separated first. And when that happens, you get a new protein, VCR ABL. Okay, and this codes for an enzyme called a tyrosine kinase. Okay, so this is an event that happens when two genes are brought closely together. You get a fusion gene and then a fusion protein. So when this happens, the cell loses control of division. What do we call that when a cell loses control of division? When a cell keeps on dividing and loses control, what's that called?
Cancer.
So this is the disease that comes from this is called chronic myelogenous leukemia. You guys have heard of leukemia, yes?
Yeah.
Yes.
Right. So this is a disease that results from this event. right? It could happen to anybody. I know a girl who's 20, now 28, didn't have a history of this in the family. She developed this, right? So, but a decade and a half ago, a company called Novartis developed a drug called Gleevec. The Gleevec blocks this enzyme. So why is this drug big step up from normal cancer treatment? Anybody know? What's normal cancer treatment? So I'm going to show you Novartis. Novartis is an important company to know, right? So Novartis is one of the largest pharmaceutical companies in the world, right? So if you look at its revenue, right? It's at $50 billion in revenue, right? These are the companies that matter, right? Amazon doesn't matter so much. Nobody cares about Amazon Prime delivery, right? These are the guys that make, for example, Ritalin, Gleevec, okay? Ritalin, I'm sure you guys have heard of, right? Voltaren, which is for arthritis, and so forth, okay? So it's a giant company. So can somebody tell me why this is a step up?
Is it easier for the body to process?
So, sorry, I can hear you.
Would it be easier for the body to process this?
Well, what's the current method of cancer treatment?
Therapy.
Sorry? Go ahead. How do you treat cancer?
Chemotherapy.
Chemotherapy, radiotherapy. So what's the problem with chemotherapy? Radiation, all that stuff.
It leaves excessive damage.
Right. So you could damage others. So what's the advantage of this guy here? It just blocks only this enzyme. This fusion enzyme. This fusion.
We wouldn't be leaving excessive damage since it's only blocking one specific enzyme.
Right. And the only one that makes this enzyme are cancer cells. So it's basically targeted therapy, right? You're targeting only the cells that make this fusion thing. That's where cancer research is going. I say this is important, right? This is important to you because you and I might know somebody that knows somebody that has a cancer, right? When you go to the doctor or when you go for treatment, these are the treatments that are these are widely available now, right? You want targeted therapy. You don't want therapy that destroys all your cells. Right? That's why I say this is important for you guys to know, right? This is common knowledge, right? No, becoming common knowledge. Is that clear?
Yeah.
Okay. So this is not just talk in a classroom, right? It's important to life in general. Okay, so chronic myelogenous leukemia. Yeah, so it's between 9 and 22. Another one is something called Ewing's sarcoma. Okay, that's between 11 and 22. Okay, this is a bone cancer. A lot of these lead to cancers, right? These translocation events. And so this one affects large bones like your thigh bone, your femur, your humerus, right? And tends to affect teenagers. Okay, no cure yet for it. Okay, so these are spontaneous events, just like the deletions. They're not inherited. Okay, so let's look at another one. Again, so now what I want to look at is duplication, right? So again, you could duplicate one gene, okay, or you could duplicate a whole bunch of genes, right? Either way, it leads to a disease. Okay, so one duplication event is duplication of the gene PLP1 gene is duplicated. Okay, this is a neuron gene, so it's an important gene for your nervous system. Okay, and when it gets duplicated, you get a disease. Okay, the rare disease, but the disease is called Elizaeus-Merzbacher disease. So this is a neuromuscular disease. Eventually, it leads to paralysis in your arms and legs, right? When this gene gets duplicated. Another one, also a neuromuscular disease, is called Charcot syndrome. There was a student of mine that actually had this syndrome at Santa Monica. Chromosome 17. This one involves a whole part of the chromosome getting duplicated, not just one gene. It's also a neuromuscular disease. Okay, so those are two neuromuscular diseases resulting from duplication. Okay, so the last kind of event that happens with chromosomes is something called inversion. So inversion, there are no real diseases associated with inversion. Inversion, the piece sort of breaks off, it sort of flips over. It's not lost like a deletion. It just reattaches itself in the opposite direction. No known diseases, right, for this, but an inversion, there's some causal relationships, right? Inversion of chromosome 9 leads to a 30% increase in miscarriages. Okay. Okay, any questions thus far? We good?
Yeah.
All right. So now I want to just talk about some other chromosomal events. Okay. So we talked about meiosis, right? So does anyone remember what the function of meiosis is?
It makes sex cells.
Makes sex cells. Very good. Okay, so I'm going to briefly go over meiosis for you. Okay, makes you egg and sperm. So in this example we saw, we start with 4 chromosomes instead of 46, one of ours. And the homologs sort of pair up here, and they separate. So you have 2 going north, 2 going south. Say by some genetic mistake, this guy that supposedly went south goes north. How many will the top cell have?
3.
3. The bottom cell is going to have 1, right? Okay, now let's say this is one of our cells. So we should have 23 guys going north, 23 guys going south. Let's say an extra one went up north. How many will the top cell have?
4.
No. So let's say this is one of our cells, right? So you have 23 going north, 23 going south. So how many will the top cell have?
23.
24,
4.
right? Right. So an extra one going up north. Right? If that extra one is, let's say, chromosome 21, then you're going to have an egg cell with 21 with 2 21s coming together with a sperm cell with 1 21. Now you're going to get 3 21s. It's Down syndrome. So when the homologs don't separate properly, that's something called non-disjunction. Okay, that can happen when you have errors in the genes involved in separating the chromosomes, mainly in meiosis and mainly in anaphase 1. Okay, so non-disjunction is the failure of homologs to separate in meiosis, usually during anaphase 1. Okay, just some genetic error. Now you can get diseases for that, like an extra 21. Okay, so if you get an extra 21, you get Down syndrome. Okay, so I'm going to show you some images of afflicted individuals, right? So kid on the extreme left has Down syndrome. So what are the symptoms of Down syndrome? So 21 is Down syndrome. What are the symptoms? Remember, you have a whole extra chromosome with hundreds of genes, right? Anybody know what the symptoms are?
I think cognitive impairment.
Yeah, cognitive impairment, very good. And yeah, and some physical problems too. And that's not the only non-disjunction disease, right? So the one on the center here, that's an extra 18. That's called Edwards syndrome. Okay, that's rarer and it's much worse than Down syndrome. They rarely make it out of infancy and the effects are much more severe. Okay. The worst is the one on the extreme right, and that's an extra 13. That's called Patau syndrome. So here, the effects are even worse than Edwards syndrome, and they almost never make it out of infancy. And the hallmark sort of symptom is also the roof of the mouth is split, cleft palate. Right? So this is the most common. You can have other non-disjunction diseases of the non-sex chromosome. Okay. Now, you can also have non-disjunction of sex chromosomes too. So remember, we talked about those guys separating, right? So you'll have 3 in the north and 1 in the south, or 24 and 22. One guy might have 1 extra one guy has 1 less, right? So, and that can happen to also sex chromosomes as well, right? So you could have something called X0, a woman with just 1 X chromosome, resulting from non-disjunction. That's called Turner syndrome. Okay, so in Turner syndrome, usually the woman is okay but sterile. A hallmark of Turner syndrome is webbing around the neck. This is Turner syndrome. So this lady has, it would be X0, 1 X chromosome. Okay, you could also be, you can have various combos of non-disjunction. You could be XXY, that's known as Klinefelter's syndrome. Usually that's a sterile male. You can have all kinds of them, XXX and so forth. But you can have non-disjunction of sex chromosomes. We clear? Questions?
All clear.
All right. So now what I want to talk about is the last chapter, let's say, or exam 3, is sex determination. Okay, it's not so fluid as you think it is in the animal world. Okay, so that's what I'm going to show. So for humans, what determines the sex, the egg or the sperm? So what determines sex in humans?
Sperm.
A sperm. So the sperm can be the X or Y, and the egg can be X. So XX is a female, XY is a male. Right? Other creatures also use the same kind of system, like roaches, and insects, right? Grasshoppers. These guys, the sperm also determines the sex. However, they don't have a Y chromosome. So the sperm could be X or nil, nothing, no sex chromosome. And the egg can only be X. So one X chromosome is a male, 2 X chromosomes is a female. They don't have a Y chromosome. Okay. Now, birds, I'm going to change the lettering of X's and Y's. I'm going to use a different letter system for sex chromosomes because birds operate a little bit differently. We're going to use the letters Z and W. For birds, the egg determines the sex, right? So the egg can be Z or W, and the sperm can only be Z. So a male is ZZ, and a female is CW, the exact opposite of humans. These are sex chromosomes in birds. Okay. So it gets sort of more weird as we go across the animal. How about social insects? Do you guys know, can you give me an example of a social insect? What's an example of a social insect? So what's an example of a social insect? Animal that hangs out in colonies.
Could it be an ant?
Okay, what's another one? Good, very good. What's another one?
A butterfly, maybe?
Butterflies don't hang out in colonies. You know the answer, guys. You guys hang out in colonies. Ants and one that has a hive.
What about a bee? Yeah.
Bees, guys. Let's go common sense, right? Ants and bees, right? Even wasps, even termites, right? So these guys don't have sex chromosomes at all. It's the number that determines this. They don't have X's or Y's or Z's or W's, right? So for them, the number determines the sex. So if they're diploid, fertilized egg, they turn into females. If they're haploid, unfertilized egg, fatherless egg, they all turn into males. Right? So if they had 32 chromosomes, let's say this was 32, the haploid number would be 16. So if they had 32 fertilized eggs, they all turn into females. If they had 16, they all turn into males. Okay? No sex chromosome, but number determines sex. Okay, for animals like turtles, crocodiles, alligators, temperature determines sex. Okay, so as a hypothetical example, if T temperature is greater than 25 degrees C, they all turn into males. T less than 25 degrees C, they all develop into females. Okay, it's not necessarily true, but this is how it sort of operates. The way they found that out is when they were trying to conserve like sea turtles, right? You take the eggs, you incubate them, and the temperature makes them all turn into males. And there goes your conservation effort. So they sort of learned, a few degrees, and it can even be tenths of a degree here and there, it changes the sex. Right? Sort of strange. Okay, any questions? We good?
Yes.
All right. So one last example. Okay, there's a type of fish. Let me show you this fish. Okay, so this fish is called the blue-headed on the Caribbean. Okay, so I'm going to show you a picture of this fish. Okay. So it exists in small colonies, and this is a picture of one member of that colony. This is, it's got a nice blue head and a nice yellow body. It's got, it's a pretty looking fish. Okay. So it comes in three varieties in the colony. Okay. There is a terminal male, which you just saw. So there's only one terminal male, blue head, and does most of the breeding and is the largest. Okay, so it's the largest fish. There's only one of them. You also have another kind of male called an intermediate male, a smaller male. It's all yellow and does some breeding. And then you have females, which are all yellow. So both the intermediate males and the females don't have a blue head. Now, remember, there's only one terminal male. If you remove that terminal male from the population, if the next largest fish, remember the terminal male is the largest, the next largest fish was a female, that female will turn into the terminal male. So what will happen is she's going to develop a blue head, and she's also going to develop male sexual organs. So we're talking about a sex change. So sex is fluid in the animal world, not static. Okay, so this is a female to male transition. It happens all, not just in fish, but even some reptile species, lizard species. Okay, this is called rotigeny.
How long does the sex change usually, like the process, long does it take to happen?
Maybe a few hours at most.
Oh, that's pretty
Less, even less. Yeah, it's freaky, right? So I'm going to show you another example, another fish, right? So you all know what this is, clownfish, right? So I just showed you one kind of transition. What do you think another kind of transition will be? So there's one kind of transition. What's another one? What do you think the other one is?
The opposite,
Sorry?
like the opposite of males to females.
Yeah, right. So clownfish exists again in small colonies. The largest fish here is a female. Remove the female, the next largest fish, if it's a male, turns into a female. Right? So this is a male to female transition. Okay, that's called protandry. Okay, so this happens a lot. Okay. Some of it is size and hormonally and timing control. Okay, any questions? We're good?
Can they continuously change like gender, or is it like a one-time thing?
It depends, right? If there's a loss of the terminal male, if an event happens, right?
Yeah.
If the event happens, yeah, the change's gonna happen.
Okay.
But the thing is, they have the ability to do it, right? Okay. So,
Sorry, Ron, but what about seahorses who have the ability to give birth? Is that any different at all? Not like directly related, but is that kind of similar?
yeah, I don't think they have the ability to change sex.
Okay, it's just
Females, yeah.
okay.
And not all of them have the ability to change sex.
Okay.
By the way, when is sex determined in humans? Are you, when you conceive, do you know your sex?
No, not immediately.
Okay, so you'll know it after 12 weeks. You're sort of default female.
Mhm.
You have a particular gene on the Y chromosome called SRY, and then you develop male organs. You head down the male pathway. It takes about 3 months. Okay. All right, so that's basically the material for exam 3, right? So mitosis, meiosis, genetics. The quiz today is on genetics. and this stuff, the sex determination. Questions? We're good? In a couple of weeks, I think, right? We all good before we move to the next topic?
Yeah.
We're good.
Okay. Now we'll talk about evolution. Okay. So evolution was sort of talked about way before Darwin, right? You have people like Lamarck. Lamarck was a French biologist, right? And he talked about the environment causing change, right? He just didn't explain the change properly. So for example, Lamarck is before Darwin. You know, Lamarck. Okay. So giraffes developed long necks because as trees got bigger and bigger, they stretched their necks, and the stretched necks were passed down to their offspring, right? So he's saying environment caused the change, but the way change was passed down was wrong, right? You don't inherit acquired characteristics. You develop muscles, you don't pass them down to your kid. Okay, so essentially, what Darwin does is he gives you a more material explanation of how this happens, right? What's happening? He didn't know about genes, so he didn't know about variation. So what islands did Darwin visit? What were the islands called?
The Galapagos.
Galapagos. And where are the Galapagos? Very good. Anybody know? Off the coast of Ecuador. It's a national park. Kind of expensive, but nice place. Okay, so he goes there and he sees bird populations with different beaks, adapted to eating different kinds of things, insects, large seeds, small seeds. He also notices iguanas. Okay, he notices iguanas that are on land and iguanas that are marine, swimming around. Okay, so the land iguanas, right? He notices, they have nice round tails, suited, for land survival. They're kind of bulky. Okay. And then he notices the marine iguanas, slightly different, more slender, and they have a flat tail. Okay. So why do they have a flat tail?
It helps them swim faster.
Yes. It helps them swim in general, right? So marine iguanas have flat tails as opposed to round tails, right? So it's an adaptation for swimming. And also iguanas tend to be more vegetarian. And so when anybody came near these marine iguanas, a salt solution came streaming out of their nostrils. And there's a reason for that. The reason is these iguanas, like I said, they're vegetarian, right? The marine ones feed on seaweed. Seaweed has a lot of salt. To get rid of this excess salt, these marine iguanas have salt glands in their nostrils, around their head region and nostrils, to get rid of excess salt out of it. So they have salt glands, right? So he notices these are adaptations for marine life. Just he notices the birds also, especially the birds and the beaks, were adaptations to different conditions, leading to different kinds of food items out there. So way before Darwin, there was an economist named Malthus. Okay, so Malthus basically stated, in simple terms, the reason we have wars, famines, all these disasters, is because there are too many people that are born. Not enough resources. So what's going to happen? Not enough resources, too many people out there, you're going to have a competition for survival. And only the ones that are best suited for that will survive. What Darwin does is he takes Malthus's concepts and applies them to all living creatures. Okay, so he says, first of all, if you look at a general population of animals, there's variation. The animals look different, just like humans, they're all different. He didn't know how this variation came about, right? He doesn't know about genes. Talking about the 1830s to 50s, right? And so the question is, okay, so you have these creatures, okay, too many of them are born, there's variation. Only those best adapted to the environment are going to survive. And if those genes are capable of being passed down, that's important because they die with you. What's the point, right? If they're inheritable, then a few generations later, you'll see a change of species. That's what evolution is, change in species over time, right? So you need some variation. You need some adaptability, right? Now, you don't develop adaptations to the environment. You either have them or you don't. If you don't, you're dead, right? So the world gets flooded, you don't grow gills. You don't have better lung capacity. You either have it or you don't. That's due to variation. We're all different. Okay? And if you do have it, you survive. And if they're inheritable, your offspring will survive and those genes get passed down. Right? So what Darwin does is he provides a mechanism for evolution, which is called natural selection. The natural selection is like the driving force, the engine that drives evolution. Okay, so this is the so-called survival of the fittest, right? Fittest, and usually does survive, so you're repeating the same thing in the same sentence. Okay, so we're going to look at examples of natural selection. But first, what we're going to look at are evidences for evolution. So what we're talking about right now is something called macroevolution. Macroevolution is basically big change, right? Macro means big. So like dinosaur to bird, that would be macroevolution. Then we're going to look at microevolution. That's where you have gene changes over time, right? From one generation to the next. So a lot of religious people, especially creationists and stuff like that, believe more, I mean, they can tolerate microevolution. Macroevolution, they have a problem, right? But there's a lot of evidence for macroevolution. Adam did not roam around with the dinosaurs, right? Okay, there's data for that. Okay, so what we're going to look at is evidences for evolution. Man was not here 65 million years ago, or his ancestors. Okay, so one line of evidence is the fossil record. So what are fossils?
There are remains of animals that like passed away or are extinct.
Right. So they could be they're like remains, or they could even be they could even be footprints. You have fossilized footprints. Okay. So as an example, as you go lower and lower into the ground, deeper into the ground, the fossils get older, right? And so this is like comparing bone structures from fossils of the common horse to its ancestors. And so you can trace back the common ancestors of the horse up to like 55 million years ago, right? Looking at their leg and hooves, right? Bones. So this is an example of a fossil record. Okay. So where did snakes come from? Snakes are relatively recent. Who are the ancestors of snakes? Anybody know? Okay, the ancestors of snakes are lizards, especially burrowing lizards. The lizards sort of burrowed into the ground, they've lost their legs. right? So if you look at the ancient snakes, the oldest snakes are your boas and pythons, right, which sort of constrict. The newer snakes are your venomous ones. They sort of bite and let you die, right? If you look at the backside of boas and pythons, the older snakes, right, you'll see you have vestigial, like these little spurs at the back. These are the so-called vestigial legs of the boas and pythons. Boa. Okay, you can still see it there from the old lizard days. Sort of leftover stuff. Okay. Another line of evidence is comparative anatomy. Okay, so here you're looking at similar bone structures, okay, between existing organisms, not like past. Okay, so for example, evolution is all about linking animals together. Okay, how do we link animals together? Right? Why do we do animal testing? Because there's obviously some similarity in the mouse or rat you're working on with you. What's the point? Okay, so if you look at a human hand, dog's leg, bird's wing, whale's flipper, and you look at the radius and all around the wrist bones, there's some similarity in the bone structure. Okay, so these are homologous, like we saw homologous chromosomes, these are homologous bone structure. And this is called comparative anatomy. It's very different from, let's say, a fly's wing and a bird's wing, right? They're both for flying, but completely different origins. Not like this. They all have different functions here, but same bone origin. Okay? So comparative anatomy is a way of also linking up organisms. Okay? What's this one? Comparative embryology. So what would this one be? What are we comparing here?
Embryos.
Yeah, this is actually, so the next slide I'm showing you is going to show you is pretty mind-blowing in terms of how we look compared to other creatures. It's real eye-opener, actually, showing you the link between humans and others. Okay, so if you look at development, so this is your human embryo. Okay, this is your fish embryo. This is your pig embryo. Do we look human here?
No.
You don't look human. Right? These over here, these little slits over here, these are gill slits. You have gill slits in the early stages of development. They develop into things like the voice box. So right now, you look like a rabbit or a pig or a turtle. Even a little later on, you still don't look more human. It's much later on that you start to look more human. That's because you have certain genes that kick on and certain genes that turn off, developmental genes. Okay, coding for certain proteins that turns a hand into a hand. Okay, for a fish into a flipper. Okay, so this is actually a very critical slide here, showing the link between organisms. Okay, any questions? We good thus far?
Yeah.
Yes.
Okay. Biogeography. Okay, this is like a vast term. Just like the geography of species. These are all evidences for evolution. So the marine iguanas and terrestrial iguanas, they have some different features, but they look similar. They look iguana-like, right? So a fox and a wolf, they're different, but they look dog-like, right? So it's sort of a comparison of species. There's some common origin that's going on. Now, one of the most important pieces of evidences for evolution is molecular biology. Okay, so this is relatively new, but it's important because now we don't need bones. Okay, we don't need fossils. We can compare based on looking at proteins or genes. So in this particular example, they're looking at hemoglobin. Hemoglobin transports oxygen in your blood. Okay. The protein is made up of amino acids. Now, if I compare a human, you and a monkey, there's 8 amino acids different. Okay, and chimp even closer, maybe like 1 amino acid difference. And compare you to a dog is 32 amino acids different between their hemoglobin and yours. Bird, 45. So you're closer to a monkey than you are to a dog, closer to a monkey and a dog than you are to a bird, and so forth. So by looking at certain proteins, hemoglobin or genes, looking at nucleotides, you can tell how close you are to that organism. You don't need to search for bones and stuff. Okay. You can also tell comparisons of human populations, looking at the DNA, which is what's done. Right. So these are just a very, very powerful tool, looking at relationships. Okay. Any questions? We're good?
Yes.
All right. So back to natural selection for a second. Okay. When we talk about natural selection, we're talking about an event that takes hundreds of thousands of millions of years. Right? It takes a long, long time. Okay. So in this particular example, the ancestral canine dog-like ancestors gave rise to the coyote, the wolf, the fox, and so forth. Right? They're all different, but they look dog-like. Right? They look canine-like, the common dog too. Okay. It's very different from something called artificial selection. Okay. So what's artificial selection?
When you choose what to breed.
Okay, so can somebody give me an example of artificial selection? Good. Yes, no?
Like purebred
So
dog.
Okay, so dog breeds, right? All your dog breeds are an example of artificial selection. So in this example of artificial selection, man is acting as the environment. So artificial selection, unlike natural selection, doesn't take hundreds of thousands of millions of years. It takes a few thousand years, much shorter period of time, right? You take a bulldog and a mastiff, you get a bull mastiff, and so forth. Okay. Now, the cat breeds too, right? Cats been domesticated over several thousand years, right? You guys have heard of the Sphinx cats?
Yeah.
What are those? You know where they are? Sort of hairless cats. These are Sphinx cats. Right? But these are clearly human bred. They don't exist like this in normal, domestic, normal cat, right? Okay, so they're bred to be without fur. You keep selecting for that trait. Okay. Okay, cattle breeds. Oops, excuse the dash. Can you have artificial selection of plants?
Yeah. Yes.
Absolutely, right? So if you look at plants, right? If you look at the common wild mustard plant, which grows in the Central America, Mexico region here, this one gave rise to all the plants you're aware of today: kale, to Brussels sprouts, cabbage, to cauliflower, to broccoli. They all came from this wild mustard plant, domesticated over several thousand years, not too long. Okay, so you got some certain traits from that. Exactly, yeah, peppers as well, right? But not from this, right? So a lot of artificial selection going on, right? All of these. Okay. All right, so back to natural selection. So what I'm going to talk about are types of natural selection. Types of natural selection. So I'm going to show you graphs of types of natural selection. Okay, so what trait am I looking at here?
Fur color.
Okay, I'm looking at fur color. Good. Okay, so these are all types of natural selection. So one type of natural selection is called stabilizing selection. So who's being favored here in stabilizing selection? Which guys are being favored? Anybody know? Yes, no? So who's being favored? Look at the crowd. Who's being favored? Keep it simple. Guys there?
Is it the dark brown or the dark color one?
So what's the range of colors here?
From very light to very kind of sort of dark.
Okay, so in the first kind over here, who's being favored?
The one in the middle is the
All right, okay, very good. That's all I'm looking for. The middle guys, the intermediate guys. Think simple. Always in biology, think simple. Think conceptually, right? So the average guys, middle guys, are favored. Okay? Right? Human babies are not too big, they're not too small. You get a sort of intermediate range of human babies that are favored. Okay, how about the next one? Who's being favored here? Direction.
From middle to dark.
Well, what's it called here? Direction, right? So you're favoring more the darker side, right? It could easily go the other way. The curve could easily go this way, and you favor the lighter side. So directional selection, you're favoring one extreme at one time. Okay. The last one, disruptive, and sometimes it's called diversifying. Who's being favored here? Last one.
Opposite ends.
Okay. So the two extremes are being favored, but at the same time. It's not so common. So it's sort of like you have some light rocks and the light guys hide out, and you have some dark rocks and dark guys hide out, and the in-between guys get picked off by predators. So both extremes favored at the same time. Clear? Yes, no?
Yeah.
Yes.
All right. So in the last century, early 1800s, okay, in England, there was a moth called the peppered moth, right? It had two alleles, coming two varieties here, a dark one and a light one. So in the mid 1800s, the light guys could hide out in the plants, and the dark guys got eaten up by birds, they got picked off. In the late 1800s, you had the Industrial Revolution. In the Industrial Revolution, the trees and all started to get dark because of the pollution. Now the light guys could hide out. Dark guys could hide out, and the light guys got picked off, like here. Okay, what kind of selection is that here?
It's directional.
Yeah, direction, right? You're favoring one direction at one time. Okay, either light or dark. Here's another example. Suppose you had a bunch of roaches in the jungle, and there's a virus that's sort of killing them. Will that virus kill every single roach?
No.
No, right? Because some are going to be resistant. And eventually, they're going to pass that resistance down to their offspring. So eventually, you're going to have resistant roaches. What kind of selection is that? If you're headed down the path of resistant roaches, which one of the three?
Would it be stabilizing?
Well, are you favoring averages here? Are you favoring any middle guys? What are you doing here? Let's think about what's happening. Who you're selecting for? Okay, so it would be directional, right? Favoring direction, right?
Yes.
Sometimes the answer could be the same things over and over again. Is that clear?
Yeah.
Yes.
All right. Okay, a couple last things. Okay, I'm going to show you examples of natural selection. And it's not all about size and you know, big assets, right? So about 11,000 years ago, there was a giant deer called the Irish elk. And he had this giant 12-foot antlers across. Now, they're not good for protection because they were too clumsy, okay? So, and too big, right? So what would be a selective advantage of having larger antlers if you're the deer? Why have bigger and bigger antlers without helping you protect yourself? So what's the selective advantage?
It's easier to kill prey, maybe.
It's a deer, it doesn't kill.
It like can protect itself better.
Okay, let's say it's not good for protection. It's too big. What's another advantage?
Makes you look like a bigger object.
But it's not good for protection because it's too big. You can't really use it. What's evolution all about? Why would you want to survive? Basic important concept here. Why do you want to survive?
To reproduce.
And that's the basic thing, right? Everybody wants to pass their genes down. Okay, humans to deer. So it could be a way of attracting mates, as I was saying, right? And passing down your genes. That's a selective advantage. Okay, so this is so-called not so obvious, but this one is even less obvious. Okay, there's a bug called the cicada. Okay, it makes noises in the forest, loud noises. So it burrows into the roots, feeds on the root juices, but it stays dormant. It comes out after 17 years. So 17 years dormancy. So it comes out in big batches after 17 years, certain species. Now predators of cicada, they have 5-year lifespans. Okay, now here's the question. So every 5 years, you got this big batch of predators coming out. The question is, why 17? Let's say it was 15 years. They come out after 15 years. How many predator life cycles is that? They come out of 15
3.
years? Yes, 3. So that means that in 15 years, I'm a bug, I come out and I'm met with this new batch of predators. But now if it's 17 years, when did the two exactly match up? After how many years will they exactly match up? Does 5 go into 17? So how many years, guys, will they exactly match up? Give me some numbers here. Yeah, hello, yeah. Well, guys, give me 1 second. Let me talk about this. Okay, so it's going to be 85 years, okay? 5 times 17. 17 is a prime number, right? So 85 years is almost a century, right? That's when they exactly match up. That's when the bugs and the predators come out. That clear?
Yes.
Okay, so in this case, time is a selective advantage. Yes, are we good?
Yes.
Yeah.
All right, so let's take a break. We'll come back in a half hour or so. I'll let you know. I don't know if my power is going to be on or not. Some people are doing some work out here. But anyway, let's give it a half hour, okay?
Okay.
Hey, guys. So one last thing about macroevolution. Okay. There are two sort of, there were two sort of competing theories, but now there's just, they sort of combined the two, and that is, one is called the gradual model, which basically change occurs gradually. And two is the punctuated model, where change, you get these quick spurts. Now how they just sort of describe macroevolution is a combination of these two, right? Overall change is very slow, but then you have some quick spurts happening throughout time. Remember, we're talking about millions of years. Okay? All right, so what I want to talk about now is microevolution, so evolution on a gene scale. Okay? So, and then I want to talk a little bit about the lab of what we're going to do with this stuff. All right. So I'm going to show you an image. Okay. So let's say we're on an isolated island. Okay. And these are iguanas. So what trait am I looking at here?
Web feet.
The web feet. Okay. So which one's dominant? This is a good review anyway, genetics.
Not webbed feet.
Okay, not web feet. Okay. So these are the homozygous dominants. These are the heteros. These are the recessives. Okay. So you're given that there are 500 animals on this island. 320 are big W, big Ws. 160s are big W, little Ws, and 20 other recessives. Now remember the genotype, that's the genetic makeup, letter code. You need two alleles, two letters. Okay, so 320 over 500, 64% are the big W, big Ws. 160 over 500 are the big W, little Ws, the heteros. That's 32%. And 20 over 500 or 4%, those are the little W, little Ws, recessive. Okay, now we want to calculate the number of alleles. Now, when I say allele is a form of a gene. That's the individual letters, the big W or the little w. So I want to know how many total big Ws and little Ws separately are floating around in that population of animals. So remember, you have 500 animals. Each animal is going to have two alleles, right? Either big W, big W, big W, little W, little W, little W. So we're going to count them all up, right? So if you multiply 2 times 500, you get 1,000. That means in that population of animals, you have 1,000 big Ws and little Ws floating around. So now we want to know what the allele frequency is, right? What's the frequency of that single letter? Or big W, what's the frequency of that single letter, the little W? So you have 64% contributed by the big W, that's 640 big Ws right there. Half the heteros, half of 32, so 160 big Ws. So add 160 plus 640, you get 800 big Ws out of 1,000 floating around in that population. And the rest are going to be little Ws. Okay, so this is generation 1. Okay, what we're going to do for the next generation is we're going to cross the two hetero guys here. Big W, little W, cross big W, little W. So let's do that cross. Okay, so big W, little W, cross big W, little W. All right, we do this cross. So what do I get in this first box?
The big W, big W.
Okay, so if I go on to the other crosses, what was the frequency of just the big W's alone, just the big letter W alone? What was the percentage? Anybody remember? Let's go back. What was the percentage of just the big W's alone?
64 for the
That's the genotype.
for the allele frequency was 800 over 1,000, so about like 80%.
Okay, very good. Okay, so as you said, 80% or 0.8. How about for the little W's?
20%.
Good. Excellent. So it's 0.8 here, 0.2 here. So if I multiply this out, 0.8 times 0.8, I get 0.64. This is the second generation. Big W, little w here, big W, little w here, so it's 0.16, 0.16, right? 0.8 times 0.2, and then the little w here, 0.04, 0.2 times 0.2. Okay, so these guys here are my second generation. Okay, do these numbers above them look familiar? Like 0.64, 0.04. Do they look familiar? These are second generations. So if you go to the first generation, look at the numbers here. Have these numbers changed as we went from generation 1 to 2? Yes, no?
Only a little bit with the 0.16 in play.
Okay, but there are two of them, right?
Yeah.
So you got 0.16 plus 0.16, that's 0.32, right? And you add them together. Yeah? Yes or no, guys? Are we following here?
Yes.
Okay. So have the numbers changed?
No.
Okay, so numbers have not changed from generation 1 to generation 2. It's 0.64 here, 0.04 here, 0.32 here. That hasn't changed. So essentially, you can model this mathematically. So there's an equation where if I let the big W's, the allele frequency, the big W's, I call them P, and the little w's, I call them Q. There's an equation, P squared plus 2PQ plus Q squared equals 1. Okay? So this is called the Hardy-Weinberg law. Okay? It states basically that no change occurs from generation to generation, right? And it assumes certain things. It's not like an ideal law. So like you have ideal gas laws, you have ideal population laws. So over here, what does P squared represent? Which term does P squared represent?
Big W?
A big W represents a genotype, P squared, right? What does Q squared represent?
Little w, little w?
Very good. And how about this middle term? 2PQ.
Big W, little w.
Okay. So these are the heteros. Homozygous dominant, homozygous recessive. Okay, like I said, it's an ideal law, population law, and assumes certain things. Okay. So since it's saying nothing happens from generation 1 to 2, okay, law assumes, or the Weinberg law assumes, the Weinberg. Okay, large populations. Okay, so you want large populations because you have small populations, greater chance for change to occur. Okay, so this is saying nothing happens. Okay, 2, no mutation. You don't have any big W's turning into little w's or little w's turning into big W's. Okay, 3, no natural selection. Okay, so this third term over here, what does that mean over here when I say no natural selection? So when I say nothing, no natural selection is happening here, what does that mean?
You don't add in predators, it's just reproduction.
So when I say it's not happening here with the webbed and non-webbed guys, what does that mean to you?
That artificial selection is taking place.
Okay, guys, I want you to think simply here. One doesn't imply the other. I'm saying natural selection is not going on in this population. You have web guys and non-web guys. What do you think conceptually? When I say there's no selection going on, what does that mean if you're webbed or non-webbed?
It's not going to change over time.
So I want you guys to think, okay, what does natural selection mean? What is the definition of natural selection? Guys, we've been talking about this for about an hour or more. What's
Is it-
natural selection?
They adapt to their environment?
So let's go back. Yeah. You need to know this for your next exam, okay, guys?
Is it the mechanism of evolution?
Okay, but what does it mean in your words? Forget my words. What does natural selection mean? It's a mechanism of evolution. And so who survives?
The animal that has better traits to its environment.
Okay.
Like maybe where like organisms better adapt like to an environment because they want to survive and produce more offspring.
Okay. So stick with those concepts that you guys just told me. All right. Now I'm saying that doesn't occur between the webbed and non-webbed guys. So what does that mean between them?
The ones that can adapt to their environment will be able to survive and produce more offspring.
Okay.
Meaning-
And I'm saying that doesn't occur between the webbed and non-webbed guys.
The webs and non-webs don't really have anything, like don't help them out. They're not really a trait that's trying to do anything.
So can you say one is better than the other?
No.
No.
Okay. Now, is that difficult to get through? That's all we're saying here. Does that make sense?
Yeah.
Yes.
Okay. This ain't quantum physics, my friends. It's not complex science here. Okay. No natural selection means I'm web, you're non-web. You don't have an advantage, I don't have an advantage. You don't have an adaptation, I don't have an adaptation. Does that make sense? That's all it is.
Yeah.
Are we good?
Yes.
No gene flow. Okay, that's the fourth concept. Okay, so what does that mean? It means, if I look at this population here, it's isolated. It means no migration. Nobody walks in or out. Because if that happens, let's say some of these guys walk in, then these numbers are going to change. So let's say these guys walk in, these numbers are going to change. So no gene flow means no migration. No migration in or out. Okay. No, which also this assumes random mating. Everybody has an equal chance of mating with everybody else. In real life, you choose your mates. You don't mate. It's not a free for all. Okay, you choose your mates. Okay, and the sixth concept, which I'm going to talk about, is no genetic drift. The genetic drift is a special concept in evolution, microevolution. Okay, there are two kinds of genetic drift. This law assumes both don't happen. Okay, one is called bottleneck effect. Okay, so let's say you have a bunch of seals in the last century, okay, and I represent them by different colored jelly beans in the bottle. And let's say there was a lot of hunting going on, so you killed off a lot of these seals, so you killed off a lot of these jelly beans, only a few jelly beans survived. These jelly beans sort of reproduce, and now you have a different population of seals, a lot more seals. Now, has diversity increased or decreased from left to right?
Decrease.
It's decreased. Okay, this is what's called a genetic bottleneck. Some disaster happens. So what's the disaster here that's happening?
Seals getting hunted.
Seals getting hunted, okay? Doesn't have to be hunting, could be like a volcano, could be an earthquake. Some disaster happens where you change the numbers. Okay, so if I go back to the original layout here, let's say a volcano or some hunting kills off 90% of these guys. Then this number is going to change dramatically. Yeah, that's the bottleneck effect. That's a kind of drift. Okay, so this law is saying this does not happen. Okay, so as an example, I'm going to show you, this is a picture I took in Northwest India. So this is a lion, an Asiatic lion. Most lions are African lions. There were only about maybe 100 or so lions at the turn of the century. So this was the lion mentioned, in the Bible. Basically, it existed in the Middle East and it was sort of exterminated by humans. There was a remnant population in India in the turn of the century, and so this is a representative of their descendants. There are about 500 left. Okay, so this is an example of a bottleneck, a drift, right? We killed off most of the Asian lions, now you have a small population. Okay. Another example of drift is something called the founder effect. So the founder effect is where a bunch of individuals, let's say some iguanas, leaves the population, the mother population, and founds a new population. A few generations later, that new population is going to be genetically different from the original population. So let's say a bunch of humans leaves this planet, it goes to another planet and founds a new population. That new population is going to be very different from the old population after some time, because they founded a new population. Okay, this law assumes that's not happening either. So you don't have any bottleneck, no disasters, and you don't have any founder effect. Sort of an ideal law. Okay, is that clear? Any questions? We're good? Yes? No?
We're good.
Good. Okay, so what I want you guys to do, okay, I'm going to show you the parts of the lab that you can do at home, but I need to see you on camera because I don't want you to like lie or anything. Okay. So you guys have this? Yes, no?
Yeah.
Okay. So these little black pieces I cut out from here and stuck them in here. So make sure you cut them out to size over here and fit them in here. Okay. This goes in here. Okay. This is agarose. It came in here, right? It's like a gel, right? Comes from seaweed. So take the cap off, microwave it for about 40 seconds, only 40 seconds. Okay. And pour it in here with this in here. Okay. Is that clear? Take
Yeah.
the cap off, agarose, it says agarose. You'll see a bubble. Observe it in the microwave and don't touch it because it's hot, right? Use something, use gloves or a towel or whatever. And it's 40 seconds, the bubble. Make sure it's all gone and pour it in here with this in. That's what hardens, like so. Right? Make sure the black strips are in here too. Yes? Before you pour it in. Okay? And let me see you do it too. Okay? And we can also extract cheek cell DNA. It's sort of an exercise. Okay? You guys have a cheek cell kit?
Are we doing
Yes?
Are we doing this now or when we meet again for the lab?
This one we should pour now and just run it now. Let's do this now and set it off. So it can harden, right? This one takes a couple hours. We're not gonna finish the lab with this. This one we can do now, the cheek cell DNA, okay, as well. Okay, so in the packet here is some Gatorade, the Gatorade packet. Gatorade has some salts and sugars. And essentially you want to make a liquid, some water and some Gatorade. Because you're going to swish it in your mouth and drink it. What it does is it's got salt in it, it's going to take off your cheek cells. Okay? And you only want to put a little bit into this cup, maybe like 5 mils at the bottom here. So that's a teeny bit down here where it says 5. That's what you want to spit out into here. Is that clear? So mix the Gatorade with some water. Pack it. Swish it in your mouth for about 30 seconds. Don't drink it, and spit out a little over here. Right? Make sense? You guys there?
Yep, makes
Yeah.
Professor, I have a question.
Um,
So my Agrace gel, I think it's spilled in the bag, like it somehow opened and it like leaked through.
This?
Yeah.
This is a gel. How can a gel leak? How do you know it leaked?
Or some sort of liquid like spilled in the bag.
Is it all empty?
It's like more than halfway.
Okay, so let's see what we can do anyway. Can you show it to me?
Yeah. Like this was the bottle and like most of it. Oh, it's kind of leaking through, but everything's just covered in it inside the bag.
Can you, it might be another bottle. Can you pull out this bottle for me?
Yeah.
The one that says Agros on it. Oh, I see. That's the buffer. Okay, so that's this one here, right? That's the buffer. Okay, but what about the smaller bottle? This one?
It's wrapped in a green bubble wrap if you are having trouble finding it.
Oh yeah, it's spilled everywhere. It's empty. It's written wet.
This one's empty too?
Yeah.
It says Agros and it's empty, really?
This one.
Or can you show me the bottle? You can take it out.
Yeah, it's. Oh, it has the gel in it, I guess.
That's the Agros.
Okay.
That's what you want to microwave. Okay, so
Would you mind showing the little box again to show how you split it up exactly?
you talking about this thing?
Yeah, yeah.
Yeah, so yeah, you want to cut out these black strips and put them here so they fit over here before you pour anything.
Mhm.
You put the little well thing here, and then you just pour the Agros. Melted Agros
40 seconds, right?
in the harden.
You said 40 seconds in the microwave?
Yeah, it'll start bubbling. You can see it, observe it. It'll start bubbling after 40 seconds. Okay.
And we cut the black strips first, you said?
Yeah, you want to cut the black strips first. Cut these, this size.
Okay, okay, thank you.
Okay, so what I want you to do, we can do this in the lab time at 12 also. Just pour it in here and we'll run it in the lab time. And the Gatorade one, just get ready for it. And I'll show you what to do in the lab. Yeah? So just,
So what do we need to do with the Gatorade one?
yeah, so what you're gonna do with the Gatorade one is you're gonna pour the packet some water like this, right? So then you just spit a little in there, right, up to 5 mils, yeah? Right, where it says 5 on this. Right? And then it comes with a little tube like this. So you open this up and you're gonna pour what you just, just gargle, and this, I mean, I didn't gargle it too much, so. Okay, and just pour it in here. That's basically your cheek cells. The cheek cells are in here, right? Okay, and then what you're gonna do, it says cell lysis solution. Cell lysis means it's like a detergent. So essentially what I'm gonna do is I'm gonna add detergent to this to sort of bust open the cheek cells, let the DNA out. Okay, so it says cell lysis, don't drink this. And this goes in here. I wanna see you guys do it anyway. Okay, the last step is then we're gonna add some alcohol, ethanol to it. And the ethanol is just going to bring the DNA out of the solution, out of your cheek cells. So you'll see it as like a white mass. That's it. Make sense?
And so we should microwave the gel right now so that it can like harden up.
So it hardens like this.
Okay.
But put the black strips in first and this in also.
Yes. Yes.
Are we good or not?
Good.
Okay, let me know if you have problems. And don't touch the bottle when your microwave is hot. You guys got the batteries, right? Set up the batteries like this too. Okay, so you have 3 batteries. This is how you want to hook them up. One on top of the 2, connecting the 2 like a bridge. So this is going to be like your power source. So eventually what you're going to do, you have the black and this is your negative electrode, this is your positive electrode. You're sort of going to put this onto the positive end. Okay. And then hook this end to the gel, the box, to the one of the black strips, and then the black one goes from negative to the box again, which we'll do. This is your power source. We're creating a positive end and negative end. Make sense?
Professor.
Yeah.
I microwaved the agarose for 40 seconds and some of it popped in the microwave, even though it was just for 40 seconds. I poured what I had remaining into the little box after I'd put the strips divider. Is that okay?
Yeah, and you put this thing in too?
Yeah, I already put that in.
Okay, yeah, that's fine.
Okay.
And yeah, no, remove the cap. Okay, you don't want the bottle to explode.
I removed the cap. It just exploded in the microwave.
No, somebody, Angel was asking me to remove the cap. Make sure the cap is off.
So we put just the glass bottle without the cap in the microwave.
Correct.
Professor, I don't have a microwave. I just have like a toaster oven.
I mean, that should work as long as this will take longer to melt, you know what I'm saying? Oh, microwave, what's wrong with you guys?
I know.
So let's see.
And then, Professor, just for clarification, so how much water do we put with the Gatorade?
20 ounces, maybe. I don't know how many. It's like half a cup, maybe like this.
So half a cup and then pour all the powder in, right?
Yeah.
Okay.
Don't swallow it. You just want to swish it around and for like 30 seconds and pour it in here. And then from here into the little tube I showed you, right? This one. Right?
And how much should we swish?
You just want to go up to 5 or 10 mils over 5 mils over here.
Okay.
This thing. And you don't want to look like a chipmunk, right? So just swish a little bit. Yeah, you could use whatever heat you want if you don't have a microwave. It doesn't matter. Just remove the cap, make sure it's melted. All you want to do is melt it. Pour everything, pour the whole gel into the thing.
And regular water is fine, right, for the Gatorade?
Yeah, sure. That's fine. Where you guys at? You gonna show me where you're at?
Professor, can you repeat how we do the cheek cell thing? I just had to run upstairs to use my microwave, but if I could just get some more clarification.
So just pour some Gatorade in a bottle, in a cup with like maybe half full of water. Swish around, a little bit in your mouth for about 30 seconds and put it in the cup so it goes up to the 5 level over here. And then take this and put it in this tube, pour it in this tube. Right? Those are your cheek cells. Basically, you're swishing around cells to put in your cheek cells.
So we just mix water and then the Gatorade powder.
Yeah.
And swish that around and until it fills up to where on the cup?
Maybe it says your cup is marked. It says 5 over here. So until the 5 level around here.
5. Okay. Got it. Thank you, Professor.
Sure. And the script should be at the bottom over here, like here. Black strips. Cut the strips from here and put it right here at the bottom. Okay. So should be the side bottom over here, side bottom over here. Okay. Can everybody also show me what they've done too? I need to see what you guys are doing. All right, you guys are good. You switched it around. And how about the others? Can I see the others? Okay, that's cool. Very good. Okay. And pour your agros in there. Okay, so so after you switch it around, I'm gonna do it the my tube. We'll run the gel later on, but okay, so we got this here, right? Now what we're gonna do is, you guys have the cell lysis solution here. It's like a pinkish solution. We're gonna pour it in here. So take this out. Okay,
Are we pouring all of that solution into here?
yeah. Let's put all of it in there. Like so. And we'll cap it. Just sort of shake it around.
Wait, what did you add inside the tube?
You mean what I just put? The cell lysis.
Yes.
It says cell lysis.
Okay, and we add all of it?
I'll add all of it, yeah.
Okay, perfect.
Just like rock it around, you know. So just so that it's like the detergent, basically is mixing with the cells, cheek cells from your mouth. Okay. And it's basically sort of busting them open, letting all the DNA out. Okay. And the last thing we're going to do is we're going to add some alcohol to it. And theoretically, it should be cold, but let's see, it should work. This is alcohol, so that ethanol on it. Okay, I'm just gonna add a little bit. Okay, and you'll see, okay, some clumps in there. Those clumps are your DNA, basically. So when you add the alcohol with your last step, you'll see clumps of DNA.
How much alcohol should we add?
Maybe a tiny bit, maybe like quarter of it.
Okay.
You'll see like sort of a clump in there. A clump is your DNA. Just look closely in there. It's pretty cool.
Oh, where do you find the alcohol?
It says ethanol.
Okay, okay. Thank you.